You are looking at two gray commas resting on a bed of ice in a supermarket. They curve in the same way, their shells glistening under fluorescent lights. But if you could shrink yourself and step inside that armor, you would discover two engineering blueprints that diverged tens of millions of years ago. Nature does not copy, it iterates. And sometimes, the difference lies not in outward shape, but in how parts are layered together like an architectural structure. To understand why shrimp are not entirely the same as prawn, we have to read the blueprint evolution left behind on every abdominal segment, every gill plate, and every tiny claw. Imagine the abdomen of a shrimp as a roof tiled by hand. The second segment overlaps both the first and the third, forming a closed structure that hugs the body tightly. This overlap is not accidental. It is a mechanical solution. The armor of crustaceans is made from chitin, a long polysaccharide composed of repeating units of N-acetylglucosamine. Picture chitin as microscopic Lego beams. When combined with proteins and minerals such as calcium carbonate, they create a material that is both rigid and flexible. In shrimp, the overlapping abdominal plates resemble tightly packed roof tiles, helping distribute force during an attack. When a predator bites down, the force does not concentrate on a single point. Instead, it slides along layered surfaces. This is a form of stress dispersion, similar to how medieval armor was assembled from many small plates to prevent catastrophic cracking. Prawns, by contrast, possess a structure more like a layered staircase. Each abdominal segment overlaps the next in a linear sequence. This produces a smoother curve, but also allows for a different kind of flexibility. The staircase design enables more pronounced expansion and movement between joints. This is not a trivial detail. It is an evolutionary statement. One design prioritizes shielding, the other prioritizes range of motion. And that difference begins with how the first bricks of the shell are laid. If we open the armor at the thorax, we encounter a sophisticated machine, the gills. But instead of getting lost in complex anatomical jargon, imagine two different systems. In prawns, the gills resemble a small tree. Branches radiate from a central axis, dividing the contact surface with water into many smaller areas. This structure is called dendrobranchiate, dendro meaning tree. Trees follow a simple principle: maximize surface area within limited space. The more leaves a tree has, the more efficient gas exchange becomes. Prawn gills work the same way. Their countless tiny branches allow dissolved oxygen in water to diffuse into the blood across a thin epithelial layer. At the molecular level, oxygen moves along a concentration gradient from areas of higher concentration in water to lower concentration in blood. Hemocyanin, a copper-containing protein rather than iron-based hemoglobin like in humans, binds oxygen and transports it throughout the body. When oxygen attaches to copper, crustacean blood appears pale blue instead of red. Shrimp, on the other hand, possess gills arranged like stacked cards in a filing drawer. The total surface area remains large, but the organization differs. Two structures. One shared goal: efficient gas exchange. Like two cities with different transportation systems that still deliver people to work on time. Nature does not choose a single path. It experiments with multiple models simultaneously. Now look at the front pairs of legs. Shrimp typically have only one prominent pair of claws. Prawns, however, often possess three well-developed clawed legs. This is not merely an aesthetic difference. It is a biological toolkit. Claws are formed from modified leg segments and function as levers. When muscles contract, force is amplified through joint mechanics. The principle is similar to the kitchen tongs you use at home. A small force at the handle generates a larger gripping force at the tip. With more clawed limbs, prawns can manipulate their environment in more complex ways, digging into sediment, gripping prey, or interacting socially with others. Shrimp, with fewer claws, often rely more heavily on speed, and their backward escape swim the tail flip. When the abdominal muscles contract forcefully, water is pushed forward, creating a reactive thrust that propels the body backward. This follows the conservation of momentum in physics. Push water in one direction, and the body moves in the opposite direction. One design favors a multifunctional toolkit. The other favors a biological jet engine. This difference is not random. It reflects two survival strategies shaped within the same underwater world. Finally, let's talk about habitat. Many prawns thrive in freshwater or brackish environments, while shrimp are more commonly associated with the salty ocean. Why? The answer lies in osmotic pressure. Imagine each cell as a city surrounded by walls. Water and salt must pass through border gates, the cell membrane. If the salt concentration outside is higher than inside, water tends to leave the cell to restore balance. If the outside environment is more dilute, water flows inward. This process is called osmosis. To survive in freshwater where salt concentration is low, prawns must constantly expel excess water while retaining essential ions like sodium and chloride. Specialized cells in the gills act like customs officers, pumping ions against their natural gradient by consuming ATP energy. Marine shrimp, living in salty environments, face the opposite problem: dehydration. They must conserve water and excrete excess salt. Two regulatory systems, two physiological strategies, like two countries with different border policies. Each species must manage its own checkpoints to survive. And it is precisely this boundary of salinity that pushed them along separate evolutionary paths. Nature never truly repeats itself. Even within a small curved body, something that might seem like nothing more than dinner evolution has etched intricate engineering choices. Layering of armor, the architecture of gills, the number of claws, and the strategies for balancing salt and water. We see two commas, but under the microscope of evolutionary history, they are two different blueprints, two structures built by the same architect known as time.